Hi, Bio 111. We have a new mitosis model that I'm going to introduce you to. I'm going to walk through and show you all the models at once and then go through each phase. Here is interphase. This is early prophase. Mid prophase. Late prophase. Metaphase. Then early anaphase. Late anaphase. Telophase and cytokinesis, and the two new daughter cells in interphase. So let's take a look. Here we have the cell in interphase. So try to get my phone to focus for you. There you go. So here you see a cell with a cell membrane, plasma membrane, nuclear membrane. D is the centrioles, F is the mitochondria, H is the Golgi apparatus, G is the endoplasmic reticulum, C is the nucleus. You can see, I'm trying to make out the letter there for you. Number 3 is the nucleolus, and that is letter A, which is the chromosome. So here we have the cell in interphase. Chromosomes have now duplicated during the DNA synthesis, or the S phase, of the cell cycle. So now we're ready to enter into mitosis. So in early prophase, we begin to see over here the centrioles beginning to create and develop the spindle fibers inside the nucleus. Again, chromosomes are now beginning to condense and form the DNA, is beginning to condense and form the functional chromosomes, which can then be divided. Coming on to mid-prophase. Here you can see that the centrioles have migrated and the spindle fibers are elongating, but we're not quite ready to attach them to the chromosomes, which have now condensed, form proper chromosomes attached to their centromeres and getting ready to be attached to the spindle fibers. So by late prophase, the cellular membrane is gone, the centrioles have migrated to poles of the cells, the spindle fibers have attached to the centromeres of the chromosomes. In metaphase, the chromosomes have now been pulled towards the midline of the cell by the spindle fibers of the centrioles, lined up ready to be separated. Coming down to early anaphase, we have now separated the chromosomes. They've pulled apart at their centromeres, and the spindle fibers are pulling the chromosomes now to the opposite poles where the centrioles are. So that's early anaphase. Really combine that in with late anaphase, because by the end of late anaphase, now the chromosomes have migrated to the poles, the spindle fibers have retracted into the centrioles, and we have truly split the chromosomal material. Then we now need to create two new daughter cells. So here we have telophase, which is when the chromosomes begin to decondense, the cellular membrane reforms, the spindle fibers break apart, organelles migrate, and we begin to get the pinching of the cytosol called cytokinesis. So that's telophase and cytokinesis. And then the last part, well, actually on to the next cell cycle, is two new daughter cells in interphase. So here you can see the two new daughter cells that look exactly like the original mother cell there. And that is the new mitosis model.